of work, and she's going to talk about some of that work with salamanders, which is how we were introduced. Just for a little bit of background, she did her undergraduate at Brown University and then went on to Ohio State, where she did her PhD and also did a postdoc with Dr. Lyle Gibbs. So if any of you are from OSU and know Dr. Gibbs, that's where Katie's genealogy comes from, her academic genealogy, pedigree, yes. And now she is just a tenured professor at Eastern Michigan University, where she teaches conservation biology and field herpetology. In fact, she was just here a couple weeks ago with her field herpetology class. And so she's going to talk to us today about the unisexual salamanders, which I don't know if any of you guys that went to Kelly's Island got to see any on the trip. You looked?
I looked.
You looked?
Anything?
Darn. Well, we'll see some cool pictures.
Some pictures.
Actually, we should have gone and we have some in the colony that we can come in between.
Maybe in the break.
Yeah, at the break, I'll bring some of our captive animals up if you want to see some live sexuals. But she's going to talk to us about gene thieves in your backyard, an introduction to the amazing unisexual salamander. So.
Thank you for that wonderful introduction. Sure, yeah, maybe a little dimmer. We'll have some pictures of some salamanders. Thank you so much for the invitation to be here. I absolutely love any excuse to talk about these crazy salamanders and my research with them. How many of you guys have ever even heard of a unisexual salamander? Okay, that's pretty good. There's at least a few in here. Explain what that means, why they're called that, and all kinds of cool things about them. So let's get on with it. Like I said, I'm going to start out sort of introducing, in general, this research system. Any of you guys know anything about normal salamanders and how they reproduce? Ish. I was figuring probably not all that many people. So what I'm going to do is I'm going to start out talking about the typical breeding salamanders that we have here in Ohio and how they are reproducing. And then I'm going to tell you about the unisexual using a mode that we call Kleptogenesis. What does klepto remind you of? Kleptomania. They're doing something that's actually totally unique in the animal kingdom as far as we know, and they're right here in our backyard. So I'll tell you about them, and then I'm just going to spend a little bit of time touching on a couple of my research questions, some projects that I did while I was at Ohio State and a postdoc funded by the Division of Wildlife, as well as some of the work I've done since moving to Michigan. Nobody booed. I said Michigan. Very surprised.
No cookies for you.
No cookies. All right. So like I said, I wanted to start out telling you about what I normal salamanders, the typical sexual reproducers that we have in our region. And I'm going to focus on 5 species. So these are all in the same genus. So they're all relatively closely related, evolutionarily speaking. When I say that these are normal or sexual reproducers, what I mean is that they have both males and females. They have only their chromosomes, so just like all of us in this room, probably, we got one set from mom, one set from dad, right? And so, to use another word for it, we have 2 sets of our chromosomes, and again, these guys are all the same. They have males. And so the key players when we're talking about the sexual species are these 5 that I'm showing you pictures of up here. We've got the Ambystoma laterale, very, very rare, state endangered in Ohio. It barely creeps into the north, into Michigan and up into Canada. And I want to point out, when I'm showing you these species, that there's a little letter. And you guys don't need to remember these scientific names. All I want you to gather is that when I start talking about genomes moving around, I'm going to use those letters to refer to which species are being stolen from, right? So much more common here in Ohio, we have Ambystoma jeffersonianum, the Jefferson salamander, common in the southern and eastern part of the state, J. We've got tiger salamanders, which will abbreviate TI, because actually we've got 2 that start with T here, unfortunately for me. Very common throughout much of North America, in fact. And then these 2 guys, which look quite similar to one another, the streamside salamander, Ambystoma barbari, and the smallmouth salamander, Ambystoma texanum. So, seen any of these salamanders in the wild ever? Yeah? Which one? Tiger, smallmouth. I already noticed some characteristics that differ among these sexual species, right? Blue-spotted salamanders have, shocking, tiger salamanders have these yellow spots. They've also got these really neat tiger eyes that are kind of gold. These 2 species are sister taxa, so they're very close relatives. They both have what call it high speeds. If you compare their faces, they basically got no snout. This guy's got a whole bunch of nose in front of its eyes. These guys basically have that really smushed little face, right? So they've all got kind of these different characteristics that we see to tell these different species apart. However, a lot of the time when you go to a wetland, you'll see something that looks like sort of a mishmash of species. So when I asked what you guys have seen when you've been out and about, some of you listed some species. If you were in Ohio, chances that you saw full species of these individuals of these species is actually kind of low. And I'm going to show you a map in just a minute of kind of where we see those 5 sexual species that I just showed you. But I wanted to spend just a moment talking about, again, sort of normal sexual reproduction in these salamanders. So all of these ambystomatids are pond breeders. They are, the whole genus are often referred to as mole salamanders because they spend a lot of the year underground. A lot of them will dig their own burrows, or they'll use mammal burrows, and they're really rare to see throughout much of the year. The time that we see them is if you happen to go to the right place at the right time, which are these lovely little, typically temporary wetlands and forested habitat. In my part of the world now, late March. As you move south, that creeps earlier. When you get all the way down to southern Ohio, it can even be late February that they're breeding. So they're waiting for these environmental cues. They're coming up out of the ground, and they are amassing in these little forested wetlands. And then, like you guys are probably, a lot of times people are more familiar with frogs. These guys have a very similar life cycle, also being amphibians. They lay eggs in the water, so they're breeding in these ponds. They're laying their eggs. Those eggs hatch out into larvae. The length of the development time of the larvae is variable depending on species, but it's usually about 6 to 8 weeks that they're cruising around fully aquatic as larvae in these little ponds. And then as the ponds are drying down, later in the summer, they're developing their limbs, they're resorbing their gills, and they're exiting those wetlands late in the summer, typically in July, where I'm doing my work. And then they grow up and become adult salamanders. So this is, again, our sort of typical sexual reproducing ambystomatid. This is what they're doing out there. So like I said, I just showed you all of those nice species and pointed out a few of their characteristics. But I mentioned that sometimes you go to some of these sites and you see something that looks like this. And this animal, I know it's a little tricky to see on the slide, but I'll first of all tell you that it was about this big, which is about the size of a tiger salamander, really big. But what color is it? It's got blue spots, like a blue-spotted salamander. It's got golden eyes, like a tiger salamander. It also has a little bit of head morphology that suggests that it may have some hexanum, that smallmouth salamander, that one that looks like it ran into a wall, right? So it's got a bunch of these characteristics that make it look like a mishmash of these different species that I showed you earlier. And we can't actually tell just looking at it exactly what it is. We have to take a little tissue sample and go back to the lab and actually look at its DNA. And when we look at the DNA of this animal down here, what we see is that it has one complete set of chromosomes, so one full genome from the blue-spotted salamander, that was laterale, that L. It's got one full set from Texanum, the smallmouth salamander, that's the T. And it's got one full set from the tiger salamander, the TI. So this is what we'll sometimes call an LP tiger for shorthand. So it's a triploid, right? We're all in here diploids. We've got 2 sets. This is an animal with 3 full sets of chromosomes cruising around, perfectly healthy, perfectly happy. It gets even a little bit more weird because if you remember from your biology class, we actually have 2 types of DNA in our cells. We have DNA in the nucleus, and we have DNA in the mitochondria, right, which is an organelle in your cell that you inherit from your mother. This that I just told you was all the nuclear DNA. The mitochondrial DNA is much more similar to Ambystoma barbari, the B, the stream-side salamander. So now we know that this animal basically has 4 different species of DNA in its body and somehow creating a fully functional organism. So this is completely crazy and really shouldn't work, but somehow does. So just to summarize this breeding system, this is, again, what we call kleptogenesis. These are gene thieves. This is actually the technical name for this mode of reproduction. They are unisexual. We call them unisexual because they're all female. So when we find these animals in the wild, we do not find males, almost ever. Very occasionally we'll find one, but we think of them as basically an all-female lineage. And how are they doing this? Well, they're breeding in those same ponds. Remember that pretty little vernal pool that I showed you? These unisexual salamanders are going there at the same time that the fully sexual species are breeding. And when the males of the sexual species are producing their spermatophores that the females pick up, these unisexuals will also pick them up. So they're basically stealing those genes from the males of the sexual species. What's really weird is that a lot of the time they pick up that spermatophore, but they don't actually use the male's DNA at all. It really just sort of triggers egg development, their own egg development, right? So they'll pick up a spermatophore, say from a blue-spotted male, but then they won't use his DNA at all, and they'll just reproduce asexually. So mom will basically clone herself. That's what happens most of the time. But sometimes, for whatever reason, a genome will be added in to the offspring. So, for example, if mom had 3 sets of chromosomes, she will produce offspring that have 4 sets of chromosomes. So what this makes, the situation that this creates is that these populations vary in what we call this ploidy, or the number of sets of chromosomes that they have, all the way from 2 sets, diploid, up through pentaploid, 5 sets. So we've actually genotyped typically juvenile larval individuals that will have 5 sets of chromosomes from any of these different sexual species. And what's really neat is that I often get questions like, is this something we did? Like, is this one of the things that we have messed up the world and created these monsters? No, this lineage is between 5 and 6 million years old. So evolutionarily speaking, it's quite an ancient lineage, particularly for this type of unisexual system, which are typically really evolutionarily short-lived. So they're very successful. They're very common. They're very widespread. And they're very weird. And I think so I just wanted to show you a quick map of these sexual species that are involved in the complex here in these various colors, and overlay the range, approximate range of the unisexual ambystoma. So you can see they've got this kind of Great Lakes distribution up through New England and parts of Canada, but they're restricted really to our part of the world, and we're kind of right at the epicenter. I often tell people that starting to work with these animals in Ohio either means that I'm a genius or completely crazy, because you may notice that Ohio is the one state where all of those sexual species overlap. So it's actually the one place where we have all 5 of these potential, what we call sperm donor species around, that the unisexuals can pick up genomes from. And so what that means is that we end up with crazy combinations of genomes in these animals. So I wanted to show you a few examples of ones. These are all either Ohio or Indiana animals. Indiana has some pretty similar biotypes, these combinations floating around. So these are, again, just a few examples. This is an individual that's a triploid. It's got a blue spotted genome and then 2 smallmouths. So you can see it's got that really kind of stubby little face, but it's still got some blue spotting, right? These are the ones that are really, really common up by me now in Michigan, because I'm in the range of the blue spotted salamander. So they start getting more blue spotted or L genomes in them. They start looking more like blue spotted salamanders, but most of ours around me are LLJ. This is the same one I showed you before, but I put another picture because they're my favorite. That's the LP tiger, found, as far as I know, only on Kelly's Island. So this is one that is fairly unique to this part of the world. Really neat, like I said, really big, kind of goofy looking animal. And then when we get further south in Ohio and east in Ohio, we start getting into the range of Jefferson salamanders. And so they start picking up more J genomes and looking more like Jefferson salamanders. This is an LJJ. So you may notice that all of these include an L. That's true for all of the unisexuals that have been identified so far. So for whatever reason, they seem to need to have one blue-spotted genome to make things work. So they all have an L. But besides that, I think that it's up to something now like 26 or 27 different genome combinations that have been identified in these animals. Like I said, all the way from 2 sets of chromosomes up through 5, although by far the most common ones are the triploids, the ones that have 3 sets of chromosomes, like the pictures that I'm showing you here on this slide. And we've got a bunch of ways of identifying kind of what's going on genetically. I don't want to get too far into the weeds on this, but I think this is a really neat picture, unless you happen to be red-green colorblind. But this is what's called a genomic in situ hybridization, where a colleague, a sort of mentor of mine did this work. This is not my work. But he was able to create species-specific probes that actually bind to the chromosomes. And so in this case, in both cases, the green chromosomes are laterale, or blue-spotted salamander, and the red ones are Jefferson salamander. So the haploid number in these guys is 14. So here you should be able, if you want, to count up 14 green and 28 red chromosomes, making that an LJJ salamander. This is the converse. This is an LLJ. So some really amazing technology that's becoming available and affordable enough to start to really do some fascinating work in terms of the genetics of this system. Well, what I'm more interested in is the ecological side of things. So what's actually going on in these ponds? How are these things functioning in the environment? How are they interacting with the sexual species? So I'm going to maybe do something a little unconventional and pause for a moment here and ask if you have any questions so far. Yes?
I know you said that it's not human-induced as hybridization, but a lot of times in nature, hybridization is brought about by environmental stressors.
Right.
Is that kind of what the best guess is as to what caused this hybridization?
So it's unclear. It's a fantastic question. The hypothesis is that based on the genomes that are involved and kind of the phylogeny of the system, that there was at some point a hybridization. It probably was in the area of Kentucky, what is now Kentucky, between a female barbari, the stream-side salamander, and a male blue-spotted salamander. So you have to remember at this point the glaciers would have kind of shoved everything down and things would be mixed up in a way that they're not right now. And so at some point there was this hybridization and likely some sort of meiosis mishap where we ended up with a triploid animal, and that's probably what set this all off.
I have another quick question.
Yeah.
So you said you mostly just see, when you see those 5N organisms,
Uh-huh.
they're usually juveniles. The more chromosomes, is it less likely that they'll survive adulthood?
Yeah, so they start to get abnormalities when they get up to pentaploid. I've gotten them as larvae. I've never gotten them as adults. We've raised some of the larvae, and you can get them to survive through metamorphosis, but oftentimes they do have some sort of like malformation of the spine and things like that. So it seems like the tetraploids do fine with 4 copies, but 5 seems to be too many. Yeah.
How long have people known that they've done this?
That's a great question. So it's actually, this is pretty new. It's been pretty recent. I would say within the last 10 to 15 years that we've really had kind of a good handle on what's going on. For a very long time, people thought that they were just normal hybrids. That you could take a blue-spotted salamander and a Jefferson salamander and breed them and end up with these things, and that's not at all what's going on. So the full species never actually hybridize. So I always try not to call these hybrids because I think that makes people think you can breed them from the other species, and that's not true. So for a long time, people thought they were just hybrids and that they were just static. So they were named species. So the most common ones are the LLJ, which was Ambystoma tremblei, the tremblay salamander, and LJJ, which was Ambystoma platinium, or the silvery salamander. And those species names are no longer accepted because now we know that an LJJ could have LLJJ offspring, right? So it's not a good species if it can change every generation like that. Chris, did you have a question?
Yeah, I think you already partially answered it from the previous question. But so when you showed that Ohio is kind of the epicenter of these ranges, if you look at it, the laterali, the L's actually doesn't come south enough to reach those other, but yet L is the one that seems to be most common.
Yeah.
So I was wondering why that happened, but it seems like it was probably because when the glaciers pushed down, that's probably where the epicenter forced those to overlap more now than they, or more then than they do.
Yeah, that's the idea, is that in that period of glaciation, those species were in contact and this hybridization happened, and that the unisexuals in Ohio basically just retained that ancestral L because they need it. But we actually see the same thing in Michigan. So you go way up in Michigan and you still get LLJ. We don't have Jefferson salamanders really in Michigan at all, but they're still sort of carrying that J northward much further out of the range of the Jefferson salamander. So it kind of goes in both directions with those genomes. I don't know who was first.
Yeah, is there, I guess, kind of ignorance here, but when I hear triploid or anything about diploids, do you think typically a sterile animal?
Nope,
well,
they are
general,
doing, in general, yeah. So
why would
and oftentimes even just diploid hybrids have problems with fertility. That's not the case with these guys. So they do have, I will say that if you're in a pond with a lot of unisexuals, you do see a lot of non-viable eggs. My hypothesis, and good data on this, is that those are actually, that they can't pick up that, they can't find a male and get a spermatophore, and so they just dump these non-viable eggs, which fail to develop.
Even the
But
males are not sterile either?
well, there are no males.
Well, you said they were, I thought.
The sexual males are normal diploids.
Okay.
Yeah, so they're fine. And the males, there's actually been some interesting behavioral work that's shown that the males will prefer to mate with a female of their own species. So they can tell the difference. So these unisexuals are somehow either having to trick these males or scavenge extra spermatophores or something in order to make this work. Yeah?
So to kind of follow up on that, are the offspring, do they lose one set of chromosomes, or do they have the same identical?
So they can be either, and actually females can have split clutches. So for example, an LLJ female up at my study site in Michigan, so a triploid female, if she picks up another L from a blue-spotted male, right, she can produce a clutch of eggs that has some LLJ triploids that are just clones of her, and she can produce, and other eggs within that same clutch might be LLLJ tetraploids. So it's not even an all or nothing thing for these females. They can produce these clutches that have multiple types of ploidy within them. Yeah, way back.
How did you observe or verify the hypothesis about them healing?
What do you mean? How do we that they're actually picking up the spermatophores?
Yeah, is that how can you verify?
How do we? That's a great question. So I've done some actual breeding experiments with these guys, but even prior to that, Jim Bogart, who did this work, had some early work trying to sort of figure out this whole system. And what you can actually do is you can capture these animals and just use like a little tiny Q-tip and swab the cloaca and test for the presence of sperm. And so what he was able to do was to do this and to look at the eggs and to show that if the animal tested negative for sperm in the cloaca, then the eggs were non-viable. So it was pretty convincing evidence that they do, in fact, even if they don't use the male's genome, for whatever reason they need either that chemical or physical cue to cause their own eggs to develop normally. Yeah.
So for like the pentaploids, they have one of each of the 5, or could they have like multiples of one?
They can have, we've never gotten one of each of the 5. That would be really cool. Yeah. No, I think that the best we've done is 4 out of 5. So, and it was from a site in Ohio where they had barbari. So it was an LTJBB, I think. That was Bogart's work, too. So I didn't phenotype that one. Thank goodness, because that would have been awful. Yeah, Tristan.
Do we know if they can reduce ploidy yet?
They must be able to, but we don't know how. Yeah. So Jim's idea is that potentially when you end up with symmetrical tetraploids, something like LLJJ, that they may undergo meiosis that creates LJ gametes, right? But we've never actually observed that. So it seems to be pretty rare because.
Well, and could they split down to even an LL, like a?
Well, right. So there's, in theory, they could, but we've also never seen that because remember that the mitochondrial DNA would be different.
Yeah.
So you could, if you got LL nuclear DNA, you'd still be able to tell if it was actually unisexual by looking at the mitochondrial DNA, and we've never seen that. And we've genotyped a lot of LLs at both nuclear and mitochondrial markers, and so far, my motto is never say never with these salamanders, but so far we've never seen it. Yeah.
So in the early stages of research, did you try and make them hybrid and see if that
The sexual species? Yes, people did. It wasn't me, but yep, absolutely. And you can, in some cases, get them to hybridize in the lab, but you have to do some things that don't happen in nature, like inject them with hormones and weirdness. So, in natural populations, it seems to be vanishingly rare. And actually, there are pretty few ponds that have a bunch of the sexual species in the same pond. Even where the ranges overlap, you often see, like out in New England, you'll see, like, very close to each other, but this pond has just blue spotteds and this pond has Jefferson, for whatever reason.
actually have one.
Oh, yeah.
So my other question was, okay, so how you're talking about they pick up the sperm or whatever, how does that work? It's like you said that they only the males prefer to breed with like their own species.
Yeah.
So
that's a fantastic question. I don't know the answer to it. I suspect that it's a combination of both. That sometimes the males are just tricked or fooled or don't care, and that sometimes the males, we know that the males produce. So we know that the males produce multiple spermatophores in a mating event. So it may be that a male blue-spotted is breeding with a female blue-spotted and makes 4 or 5 spermatophores, and that female blue-spotted may only pick up a couple. So it's a really interesting question, and people call them, and I call them because it's funny, gene thieves, and we talk about them stealing, but I wonder if they're more just scavenging, and I suspect that's probably more commonly the case, but that's just the hypothesis. I'm making things up. Yeah.
Is there a theory as to why there are some mole salamander species that you don't find, like the marbled and spotted salamanders that you don't
That's
find?
a good question. I took out the slide that talked about that because I was trying not to say too many things. But yeah, we have two common ambistomatids that are not involved in the complex. The marbled salamander, world's cutest salamander, they breed in the fall. So there actually are one ambistomatid that has a different phenology for the breeding. They breed in this part of the world, well, you gotta go down to southern Ohio, but in September. And they're actually breeding terrestrially and hanging out in areas and laying eggs terrestrially, but in depressional areas where wetlands will form. So it's a really neat life cycle, but they're just breeding at a different time of year. The yellow-spotted Ambistoma maculatum, we don't know. They're breeding in the same ponds at the same time of year. They just seem to be genetically incompatible. Yeah, so we do have a couple of sites actually where we have what looks like unisexuals using spotted salamander spermatophores to reproduce only asexually, but we never see those genomes getting incorporated. It doesn't seem to work. Yeah.
Can you have like three of the same kind, like L, L, and L?
So far, no. Nope. So far it looks like the rules are they need at least one L and at least one of something else. Yeah. These are such good questions. I just want to take questions all day. Okay, then I'm going to run out of time. Yeah.
You believe that the first one was an L and J combination?
LB, actually.
LB.
Mm-hmm.
What leads you to believe it was an L and a B?
So the B, we could be fairly certain that the barbaride, the female involved in that was a barbaride because the mitochondrial DNA in these unisexuals, right, is maternally inherited. And so they all have very similar mitochondrial DNA, and it's all very similar to barbaride. So there have been obviously some changes in the intervening 5 million years, but of all of those ambistomatids, it looks most like barbaride. So we're pretty sure that unisexual Eve was a barbaride. And then because they all require that L, it seems like probably the male involved was a blue-spotted. But it's, again, something that's probable but not for certain. Good? Those are such good questions. Yay. Okay, so how much time do I have, like 10 minutes?
Sure.
Okay. Okay, so I'm going to skip some of the Michigan stuff and just tell you about the Ohio stuff quickly. So one of the things I was really interested in investigating during my postdoc was whether these things are, we often treat them as just kind of this bulk, the unisexual complex, right, as if they're just this thing. And I wanted to know if that was reasonable or if they're actually sort of ecologically different from one another and from the sexual species that they're most genetically similar to. And so what we did was this crazy sample palooza across Ohio. I ended up with 61 sites across the state where I basically just tried to go to any place that we had any ambistomatids breeding and sample randomly from that population and see what was there. And we took a whole bunch of environmental data as well. We ended up with over 1,300 salamanders sampled over the course of this project and genotyped them using a panel of SNPs, or single nucleotide polymorphisms, which we identified, which have particular locations in the genome where I knew, for example, full Jefferson salamanders would have a T, so I'd get this red peak. Full blue spotteds would have an A, so I would get this green peak when I look at that data. And lo and behold, when I look at a unisexual, there are both peaks. And we could actually use the ratio of those peaks to figure out how many copies of those genomes were present in those animals. And so this was a pattern across all of Ohio. So we had a really strong sort of gradient from the sort of unglaciated part of Ohio up through the Northwest, where when you go down to southern or far eastern Ohio, you really don't find unisexuals. You're really into the range of full Jefferson salamanders, which are the red locations on this map. And as you move north and west, you get higher and higher and higher frequencies of unisexuals, which is the gray. So the yellow here is Texanum. This is the smallmouth salamander, so they fall in here in the middle and western part of the state. And then as you get up north, and particularly if you move further north into Michigan, you start finding more and more blue-spotted salamanders, which is this lone, I think I had one site with actual full blue-spotted salamanders in northwest Ohio. So that was interesting, but we wanted to know really also how these species were sort of splitting the ecological space with the sexual taxa, right? And so I wanted to compare a sexual species with its most similar unisexual, so the Jeffersons versus the LJJs, for example. So this was a map where I had either or both of those types of salamanders, I'll say. And I'm not going to go through any of the statistics or the other environmental data that we took and how we did the analysis. I'm just going to show you that the most important thing as far as all of the environmental data was this metric we looked at called landscape resistance, which basically just summarized what was going on within 1 kilometer of the pond. So how much of that surrounding landscape was made up of something that we would call low resistance for a salamander, like forest, versus something that we would consider to be higher resistance, like agriculture. And so we could assign these resistance values based on a bunch of other literature and come up with a value that basically just summarized, like I said, what's going on in that sort of broader landscape context around that pond. And this is the variable that turned out to have the most explanatory power for all of the stuff we looked at. So this was that Jeffersons versus LJJ comparison. And what was interesting here is that we saw a statistically significant difference for sites where we had just LJJ unisexuals versus any sites where we had full Jefferson salamanders. The lower numbers here, the lower resistance values, are what you can think of as nicer ponds, right? They have more forested habitat surrounding them, less human disturbance, things like agriculture and residential development. So, I don't know what that means. I don't want to connect to the server at this moment. So that was the pattern with the Jeffersons versus the LJJs. Basically, the Jeffersons were in those higher quality environments, and the LJJs were in the environments that we consider to be perhaps more degraded or more human impacted. So that's neat. So then we decided to look at Texanum versus LTT, which is the other common unisexual that we have here in Ohio. And so they were, again, distributed sort of further north and west in the state. And this is the problem. When you find some cool pattern and you decide to keep delving into your data, sometimes you find the exact opposite pattern. So for these guys, in fact, those sites that we found Texanum only tended to have the highest landscape resistance. So remember, with the Jeffersons, the fully sexual was in the nice habitat. Here, the unisexuals are in the nicer habitat, and the full Texanum are in the kind of crummy and degraded habitat, right? So it was exactly the opposite. So what does it mean? Well, it's up for interpretation, right? We have sort of two ways of thinking about this. Perhaps the Jefferson salamanders are outcompeting the unisexuals for that good habitat. Perhaps they're able to push them out. They're better competitors. The alternative is that perhaps the LJJs are just tougher, right? Those unisexuals are just able to use the sort of more degraded habitat that the Jeffersons aren't able to take advantage of. So it could be one, it could be the other. We won't know unless we're able to do some more experimental analysis. Either way, the relationship is the exact opposite for the Texanum, the smallmouth salamander, and its most closely associated unisexual. So the one thing we can conclude is that these organisms don't seem to be all the same, right? They're definitely using different types of habitat and potentially interacting with those sexual species in different ways in different parts of the state. And our sort of current understanding of this data set is that perhaps what we're actually seeing is that if there's kind of this spectrum of habitat quality from high to low, maybe we just have them kind of arrayed like this, where the Jefferson salamanders are using that kind of highest quality, most intact habitat, followed by LJJ, followed by LTT, and then Texanum. So that when we just looked at them pairwise, we saw a reversal in that relationship, right? When in fact, maybe we're just kind of looking at different subsets of this whole continuum. So, all right, I'm going to skip the Michigan stuff, and I'm happy to answer questions about it later, but I don't want to take up all of your time. I'll just mention that I think the biggest question in my mind, and because I can't give a talk without showing Jim Bogart's fantastic cartoon here, is people always ask me, well, are they doing this on purpose, right? Why are they sometimes adding genomes and sometimes not? I think that's the most fantastic question and the most fascinating question about this set of organisms, because if you think about it, if they can just wander into a new pond and grab a whole set of chromosomes from that potentially locally adapted species for their offspring, that's like this amazing evolutionary fast track, right? And the fact that we see them, they're so widespread and they're so common, I didn't mention, but a lot of those northern Ohio and southern Michigan sites, these guys are over 90% of the salamanders in the pond. So they're super, super common. They're very successful where they exist. And so I suspect that they're doing this in an adaptive way. And what I mentioned, I've been doing some breeding experiments to look at this exact question. I thought I had a picture of my little breeding boxes, but I don't. But we're basically putting them in boxes with different types of males and collecting the eggs and seeing kind of when do the females use the male's genome versus when do they not. Is it just temperature dependent, which we know there is a temperature component, or are there other ways they're sort of making this decision? I use the word decision loosely, but I suspect that we may see some really neat patterns with these breeding experiments. So stay tuned. And with that, I'll just thank everyone that collected samples for all that Ohio. That was a lot of samples. I didn't collect all of them. And funding agencies, Ohio Division of Wildlife funded most of that work, so thanks especially to them. And if I have another minute, I'll take another question. If not, have a cookie.
No, please.
Yeah.
So if there's not a sexual male present, they only have non-viable eggs, correct?
Right.
Okay. What's the sexual species that don't tell you that?
That's a good question. So we've gotten full tigers out there. So they may just be using the tiger genome. I don't think we've ever gotten full taxonomy. Do you remember? I don't think Jim's ever gotten one. So a lot of the ones out on Kelly's are LTT. So they look, and some of them look just like a full taxonomy. But I don't think we've ever gotten a full smallmouth out there. So as far as what I've actually genotyped, it's just the tigers.
And the tigers,
and they're few and far between.
we only see them at the state park sites, the full males. I think that the ones we've only, not that
True.
Jim has done.
There are marbled salamanders out there, but again, they're breeding at a different time of year. So, so it's a mystery, and there are a lot of sites we have, particularly in this part of the world. It seems like Northwest Ohio, Southeast Michigan is kind of the peak frequency of these guys, and there are a lot of ponds where I have sampled 60, 80 animals and never gotten a full sexual animal. They're all unisexual. So how are they persisting there? Your guess is as good as mine.
Story continues.
Yeah.
So can these females find or know the difference between the different spermatotypes? So if they see an L or a J, they go, I want that J, not the L. Or if you make
I would love to try that experiment. I don't know. Like I said, they're typically in a pond with just one of the male types.
okay.
But you could do some experimental work to look at that question and see if there's a preference there. I suspect that they are either actually engaged in courtship with the male whose spermatophore they pick up, or they're right there while the male is breeding with. So I suspect they're probably skewing in on the male rather than just the spermatophore, but I don't know.
So if they're mating with a J and they go, oh, this guy's a J, I don't want him, I just want to be able to make an egg. It's not going to be fertile, but
Well, they still will produce fertile eggs then. So if they mate, but they don't want to include the male's genome, they don't have to,
okay.
right? They just then would produce clonal offspring.
Okay, and then I guess my question is, do they know that what they're getting into?
I wish I knew. Yeah, I don't know. So the Michigan stuff I skipped shows that there's definitely, temperature plays a role in whether or not that male's genome gets added. But we wonder if there are other things going on too. And I suspect there are, particularly because we see so many of these mixed clutches where some of the offspring have the male's genome and others don't. Sometimes they all have the male's genome, sometimes none of them have the male's genome. So it's clearly a flexible trait that has been under selection for 5 million years. So I'd be surprised if there was really no pattern. Yeah.
This may be a silly question, but like you said, you never say nothing's possible with these salamanders. Are the male spermatophores, if they're not taking the genetic material from them, are they reusable for like a female of the same species?
Well, they pick them up. There's internal fertilization in these guys, so they're actually picking them up in their cloaca. So I would guess not, because they're picking up the whole thing. There's really kind of nothing left after that. But it's an interesting question. Yeah, and we don't know what that cue is either. If there's some suspicion that it's actually just physical. So with some frogs, developmental biologists, people can get frog eggs to develop just by pricking them with a tiny little pin, and that physical stimulus triggers development. So I'm dying to try that. Yeah.
have the ability to basically evolutionarily fast-track themselves by some of those nearby species, but they need the nearby species to survive. So do they run the risk of outcompeting nearby species and therefore driving themselves into extinction?
Fantastic question. Yeah, so the studies that I'm working on in Michigan, I'm hoping to get some really long-term data to look at exactly this. So what I suspect is that, yeah, if they become too common and the population is too male-limited, they will, in fact, sort of drive themselves, potentially probably not to local extinction, but to lower numbers, right? And then until the males are either recolonizing from elsewhere or that sexual population builds itself back up. So if we could watch these guys long enough, maybe we see some sort of cyclical dynamics with these sexual species, but no one has done a sort of long-term mercury capture work like the part I get from inside that done. Yeah, wave back. Stop me whenever you do that.
I'm just kind of a little confused by this, but like, what's the overall point of the unisexuals capturing the genotype? Is there like an environmental factor that they need to adapt to, or like?
That's a great question. So are there, there are probably different local factors and climatic factors and things like that for which different genomes are better adapted. So we know, for example, that the blue-spotted salamander has the most northern distributed range. So if you're going to be a unisexual in Michigan, you probably want to get a bunch of blue-spotted genomes so that you can handle that cold, right? And so yes, there do appear to be. There certainly are niche differences among all of those sexual taxa, and so the unisexuals living within those ranges start to closely resemble those sexual species that are living there. Does that answer your question?
Yeah.
Yeah.
Is there like more common one and then like more rare species, or does it vary on what place you're in as which is more common?
Yeah, it depends. So range-wise, the most common are LLJ if you're in the northern part, and LJJ if you're in the southern part. LTT is also pretty common here in Ohio. But then, so those are for sure the most common across the whole unisexual range. But yeah, if you are up in Michigan, you're pretty much just going to find LLJ and LLLJ and LLLJ because that they're with blue-spotted salamanders, so they're just picking up those genomes. So it just depends who they're around, who else is breeding in those ponds with them.
So is there like one like super rare one, I guess?
Yeah, so the Kelly's Island ones, I would say, would be, they're, so far I've only ever found them out there. I don't know anyone that's found them anywhere else that have those tiger genomes involved. So that's one. You know, they're up by northeast Ohio. I have a few sites where we would get LTJ. And then in southwest Ohio, we start to get some with barbari, with the bee involved. So there's actually only one site I'm aware of down near Cincinnati where you get barbari in there. So there are certain locations where we have some really weird combinations that are not found elsewhere.
So the chromosome picture you put up.
Yeah.
Maybe it's just the stage when that image was taken. It didn't appear like there was a lot of chiasmata going on, or was it at the stage of meiosis? So it didn't seem like there was a lot of crossing over.
Yeah.
Either. So how did that muddy the water? I saw in the subset box you had it, but it didn't seem
I don't know.
The
That's
whole
Jim's
swab.
work, so I'm not exactly sure how he creates those chromosome smears and what exactly. I'd have to ask him. But yeah, he does see crossing over, not infrequently.
Okay.
So that happens.
If we could thank Kate for the presentation. We'll start the introduction. So the cookies are in the back, and then we'll start the introduction right around 8 o'clock with Fossil giving a little introduction to what that. Absolutely my pleasure to introduce our guest lecturer tonight. So we have with us Chief Scott Zody from the Ohio Department of Natural Resources Division of Wildlife. Scott's a lifelong resident of Ohio. He actually grew up in, I think, Wayne County is where you're from. Tri-State? Tri-Way High School, and then an Ashland graduate from college. Been public service for 25 years. 17 of that was with the DNR. You've been anywhere. I know you were a legislative liaison, deputy director, assistant director, even a spell for a couple months as director. Yes. Director. So now the chief. I know he's a Buckeyes fan. I know he's an Indians fan. Unfortunately, I saw he's a Bengals fan is the thing that I saw too. There's a little cheer in the back. I'm a Cleveland homeboy myself. But as chief, basically, his role is to basically monitor and administer, the wildlife in Ohio. He's the individual that's going to be responsible for your hunting, your fishing, and your trapping license. He's going to be the one that works on these endangered or threatened animals in the state and managing this resource. And just like Sea Grant and Stone Lab, a big part of his job is the outreach to educate the public. And so he spends a lot of time working to see that agency is educating the public that's out there. So I don't want to take too much more time from Chief Zody, but we do ask if you have a couple minutes of this, and you spoke for us before up here, if you could kind of take maybe the first couple minutes just to talk to students about what path you kind of took to get where you're at. So it's really interesting for for these students to figure out when you're meeting the chief of the Division of Wildlife up here. This is a fantastic guest speaker. So if you're sitting there, how do I get to be something like the chief, or what kind of roads and paths might you want to take? So we'll take that.
They're gonna be really disappointed.
I don't want to tell my story either. You have to live in the gutter for a while, and you have to. No, so if you could just give like a quick introduction on that, and then go into questions.
Yeah, absolutely. Thanks, Chris. I appreciate it, and thanks for having me back again. I really enjoy the opportunity to get up here and speak to Stone Lab. And as Chris said, I've kind of bounced around in several different opportunities and several different roles within ODNR. So it's going to tell you one of two things. Either I've had a really interesting and varied career, or I'm a damn fool that can't hold a steady job. So if you ask my wife, she'll say one thing. If you ask my daughter, she'll tell you the other thing. So figure that out. No, I actually got to the Chief of Division of Wildlife by a rather indirect path. I think, at least in recent modern memory, probably going back the last 40, 50 years, the first chief of the Division of Wildlife who did not work his way up through the ranks of the division. I actually came, as Chris said, from other roles in state government and within DNR. But I did, I guess this is kind of indirectly related. My minor in college was geology, so I did start a little bit down the path of a DNR career. But my major was actually history and political science. And so I kind of in a roundabout way ended up, like I said, started in the legislature as an aide, came up to DNR, started as legislative liaison, which quite frankly, to me, that's the one job in ODNR where you learn about the whole department and probably prepared me for all of the other roles that I've been in throughout my career. But in working as a deputy director, I focused on the recreation side of the house, which was state parks, wildlife, forestry, natural areas and preserves, and things like that. So I did pick up some indirect experience working on wildlife issues. I am a late blooming hunter. I've been a lifelong angler. Started fishing as a wee kid. Didn't actually get into hunting until I was in my 20s, and an uncle kind of took me under his wing and took me out. So I'm kind of an exception to the rule from that standpoint as well. So yeah, I'm very, I guess you call me the non-conforming chief. But again, I guess that means you don't have to necessarily be a wildlife biology major to end up in that position. It's all a matter, I think, of being in the right places at the right time, but also staying focused on what your goals and objectives are. So there's your lesson for the day on strange career paths. So for Division of Wildlife, what do we do? Obviously, we manage Ohio's native wildlife and its habitats, enforce fish and game laws, as Chris mentioned, but then education, outreach, and then, also I would include under wildlife education, outreach, research. Some of the work that Kate does. I mean, that's really one of the neat things is being able to be here tonight and listen and see how some of our dollars are being invested in some of the really cool stuff that it gets to produce and results that we see. Our mission, conserve and improve wildlife resources and their habitats for sustainable use and appreciation for all. And I think that's a critical piece, and we're going to get to a little bit more of that in this presentation. Just talking about our 5 cornerstones with what we do and how we organize ourselves from day to day and year to year. Again, stewardship, how we take care of those resources. Opportunities. Where do we look for partnerships to work with other folks, whether it's Ohio State, whether it's Stone Lab, whether it's non-government entities like, National Wild Turkey Federation, Pheasants Forever, things like that, the connections connecting to our public. It's not just, hunters, anglers, and trappers, but it's all of the public that we serve, but at the same time, observing and protecting and trying to carry on our traditions. As Americans, as hunters, anglers, trappers, those are very important to us to continue to have those traditions, and then always, obviously, we all strive for excellence. How we're organized, we have 5 district offices around the state. We have we're budgeted for 425 employees. Right now, we're actually about 400. We're kind of playing a little bit of catch-up because we had a little bit of turnover the last couple of years. I won't bore you with a whole lot of those details, but getting into where we're organized, the 3 fisheries research stations, we have Sandusky office, we have an office in Fairport Harbor, and then our inland fisheries unit is actually down at Hebron, which is down by Buckeye Lake in central Ohio. 2 wildlife research stations. We own, control, or administer approximately 200,000 acres of public hunting and wildlife areas, and then we have 6 fish hatcheries scattered around the state, and obviously the one over here across the bay that we're partnering with Stone Lab on used to be a Division of Wildlife fish hatchery many moons ago. But it's really cool to see what's happened with that and how it's evolved as an education center. So some things we're going to talk about tonight. I want to talk about the North American model for wildlife conservation, a little history, a little about Ohio, key legislation that was involved in that. Also touch on a few hot topics of what's going on around the state and what we're getting our fingers immersed in here most recently. North American model of wildlife conservation. This is quite frankly very, very unique throughout the world. When you look at the history of the country and you look at how wildlife and fish resources were managed or not managed in many instances in other countries and on other continents, we were kind of ahead of we are way ahead of the game when you're talking about having a passion and having a belief in wildlife as a resource that needs to be sustained, protected, and utilized properly. So the pillars of the model, they belong to all citizens. It's a public trust. It's managed for sustainable use. No individual owns it. The state doesn't own it. But the state is responsible for managing those resources on behalf of you and all the people in Ohio. Limit commercialization, democratic law and legal process. That's got some history here in this country. Equal hunting opportunities, privileges to hunt and fish. In other words, this isn't, the days of Sherwood Forest and Robin Hood where only the nobles were allowed to kill the king's deer. Anyone can have the opportunity to hunt and fish. User pay, public benefit. That's huge because it's the hunters, anglers, and trappers who are actually, through their license fees, through the excise taxes that we'll get into here in a little bit, provide the funding for the fish and wildlife management and habitat sustainable use. But the entire public, even non-hunting public, I don't care if you hunt, with a camera or a gun or binoculars, you all benefit. Wildlife is treated as an international resource, and we base our decisions on the best available scientific and biologically available data and information. So we try to keep the emotions and keep the political considerations. Funding, history, just a little bit. Obviously, the state budget is where most of our funding is appropriated, but most of our funding dollars come from either hunting licenses, fishing licenses, and federal funding that was initiated in 1937. Federal funding, there are 3 major legs of the stool. The Pittman-Robertson Act, or what we refer to as PR, Dingell-Johnson, or what we call DJ, and state wildlife grants, which we affectionately call SWIG. Like the college students, you know what I mean. But those 3 funds provide roughly average about $16 million a year for support and programs in Ohio, and actually in recent years it's actually been even more than that. I'm not going to dwell a whole lot on this, but this is what we call our cycle of success for fish and wildlife management when you talk about from the funding standpoint, where the hunters and anglers make purchases, whether it's a gun, a bow, a fishing pole, a license, and those funds go to the feds, to the Fish and Wildlife Service. They're apportioned back to the states. We match those down for specific projects for fish and wildlife habitat and use. Benefits the habitat, benefits the sportsmen, and then as a result we have better hunting and fishing, and the cycle continues. PR, just talk a little bit about that. It's an excise tax on the manufacturers of guns and ammunition, and then archery equipment was actually added in the 1980s. The amount it produces a year, it's a 75-25 reimbursable match, which means that I, as the Division of Wildlife, if I've got $1 million project and it qualifies for PR aid, I have to spend that $1 million up front. Then I apply to the Fish and Wildlife Service. I get 75% of that money back, and that's where the excise taxes that are paid nationwide go to DC. They apportion them back, and they say, well, this is what you're eligible for. Okay, so now I got to spend those dollars and make sure I match them down. Again, it can be for management. It can also be for research. And this is focused on terrestrial animals and wild birds. And one of the key elements of both PR and DJ, when the founders or the sponsors passed this legislation back in 1937, they had incredible vision, because they said, we are going to require that these dollars can only be used for the administration and management by the state fish and wildlife agencies. You cannot divert them to any other use. And it's really helped many states keep their funding focused where it's supposed to be and not be diverted off into other things. Not to say other states haven't tried, but the feds keep a pretty good eye on that. This is just kind of a breakdown of where those excise taxes come from that are paid into PR. Again, guns, ammo, archery. DJ, or on the fish's side, again, it's an excise tax on the equipment and tackle, rods, reels, lures, you name it. It also includes a small motorboat fuel, and there's the excise tax is applied to certain classes of outboard engines that go on small fishing boats. Again, 75-25 match, again, has that diversion language right in there. DJ was passed in the early 50s. It took a little while after PR went through to a couple, little thing called Depression and Second World War kind of got in the way of things. So it took a little longer to get that act through Congress, but it's there, and here's how those revenues come in and how they're categorized. And then lastly, state wildlife grants, or SWIG. One of the thing that was kind of missing from our funding stool, you had fisheries and aquatic, you had terrestrial wildlife and birds, but there wasn't anything there really for endangered or threatened animals, or for things that weren't necessarily of a consumptive nature, like a salamander. So the Congress, after several years of lobbying effort by both state agencies and non-government organizations like TNC, birding groups, and things like that, we're trying to create a dedicated funding source similar to PRDJ. Congress finally said, okay, look, enough. We really don't want to go down that road to permanently create anything, but we're going to create these state wildlife grants, and we'll provide funding for species of greatest conservation concern and try to provide a amount from year to year in that regard. So we usually get $1 to $2 million a year. The match has varied over the years. I think right now it's 65-35, which is kind of in the middle, which is great. But we typically use, people are aware of income tax checkoff for wildlife. Okay, you're aware of that. We got license plates that we provide sales for, and you can also do donations. Most of your income tax donations are used to match those dollars down and put them into on-the-ground projects. I'm not going to spend a whole lot of time on this, but basically our biggest challenge right now is recruitment retention. We need more hunters and anglers, and we've been slowly bleeding numbers, especially on the hunting side, for the past several years. Obviously, since our hunters and anglers form the basis of our funding, it's like any other smart business. If you want to stay financially stable, you better find more customers. So it's one of the things we're going to be focused on now and into the future. So, oh good, I did hear a couple of chuckles. I was going to say, I hope, I'm not that old, and I hope people will recognize who that is. Current issues, especially here, obviously one of the big concerns we're battling right now is invasive species, in particular on the aquatic side. We know we've got zebra and quagga mussels and gobies and white perch. Obviously the big one is Asian carp. We've been working very closely with the Asian Carp Regional Coordinating Committee, which is focused on the Chicago area waterway system and trying to eventually, maybe in my lifetime, hydrologically separate the Lake Michigan from the Mississippi River drainage basin, which is the greatest threat to having those fish move up the Illinois River and get into the Great Lakes system. But we also have Asian carp moving up the Ohio River. They've been found individually and in small groups above Louisville, around the Cincinnati area, and then individually, singly further up. They've actually found individuals as far up as almost to Pennsylvania in the Ohio River. So obviously that's a concern because we've got two fronts. We've got Chicago into the lakes, and then we've got up the river. We actually have 4 connections in Ohio where at times the waters of the Ohio watershed, the Lake Erie watershed and the Ohio River watershed come together. We've got Grand Lake St. Mary's, which is an artificially created man-made lake that was built to feed the canals. It actually has 2 spillways, one at the west end that goes into the Wabash River, one at the east end that goes into the Ohio Erie Canal and drains into the Maumee River, which comes into Lake Erie. So it's not a high risk, but it is still a risk for passage, maybe not of Asian carp, but other aquatic invasive species. We've got Little Killbuck Creek, where if the floods come up high enough, kind of like what they're doing now, you can connect the Killbuck Creek and Lake Erie watershed, and then over in Akron, one of the old connections with the Ohio Erie Canal and the Tuscaloosa River come together. Again, there's potential there, and then over at Mosquito Lake. So that's we're working on closing our connections here in Ohio. But the question of invasive species is constant, and we're actually working bait dealers to make sure that if they're bringing bait in from out of state, especially down from the southern states, that they're bringing in the right kind of minnows, and they're not bringing in big head or silver carp minnows, because it's pretty amazing how closely a fathead resembles an Asian carp minnow. So we got to keep a close eye on that sort of thing. Energy development. Obviously, there's a lot of work going on in eastern Ohio with the shale boom. We are not doing any kind of leasing or drilling on state-owned lands. Don't know where that's going to go at any point in time, but right now we're kind of more keeping an eye on things and working with other public and private landowners on evaluating potential for whether it's forest fragmentation, whether it's any type of pollution issues that may or may not come up. Right now they're doing pretty good, but there's always a concern about once you start putting a bunch of pipelines in, depending on how you locate, where you locate, and how those things are installed, you want to make sure you're not having some detrimental effects on your forest wildlife-dependent resources. Algal blooms and fisheries, obviously we're paying real close attention to what's going on up here in Lake Erie. We're also spending a lot of effort at Grand Lake St. Mary's, which has had some really bad blooms in the past 5 or 6 years. But we partner with OSU, the Aquatic Ecology Lab, and their Terrestrial Ecology Lab, as well as working with Chris and the folks up here. We've got gathering samples, analyzing water, and we just, one of the grants that was just released through the Board of Regents to OSU is going to be looking at fish tissue testing and whether or not there's any type of bioaccumulation that is occurring with respect to fish and harmful algal blooms. What we've seen so far in testing fish, both at Grand Lake St. Mary's as well as here in Lake Erie, is that is not occurring. The fish swim through the algae. Yes, you can find traces in liver and internal organs, But as far as taking up the any type of microcystin in the fillets itself, that is not happening, and that's good. That's good news from our standpoint. But we're going to keep a close eye on that. Bats and monarchs. Anybody's been paying attention at all? We've got the northern long-eared bat, which is now a federally threatened species, and under what's referred to as a 4D rule. So you add that to the Indiana bat. There's probably little browns and tricolored bats that are probably going to be shortly under consideration and possibly listed. This impacts all sorts of different areas in the state, from both a private sector as well as business, whether it's wind energy, potentially other types of development projects, because once you get something on the threatened and endangered list, then there's all sorts of hoops and things you need to be careful of. And then monarch butterflies. Monarchs are in pretty serious decline, and a lot of that has to do with loss of habitat up here in North America. For a while, they were concerned because the wintering grounds where it would go down to Mexico were being heavily deforested. They've kind of put a stop to that and turned the corner on that, and have got their habitat down there turned around. But the number one thing that a monarch butterfly needs is milkweed. And milkweed is not considered very popular to have around the farm. So we're trying to find a way to balance the wise use and the needs of the agriculture community with the needs of the monarch. I mean, I can remember, in the not-too-distant past, monarchs were a dime a dozen. You'd see them anywhere and everywhere. And now I got to thinking about it here in the past year or two. I can't remember the last time I saw a monarch, which is pretty scary, considering how popular or how populous those things have been, especially in Ohio. So we're working with Fish and Wildlife Service, other states, doing some partnering with NGOs. We actually got a really neat project we did last year in cooperation with the State Department of Transportation, Pheasants Forever, and our staff down in Southwest Ohio. We actually worked with ODOT, identified some excess unused right-of-way adjacent to some roads that they had down in Southwest Ohio. We said, gee, guys, you got this land here, it's grass, pain in the butt to mow it, to cost you money. You guys mind if we plant some habitat in there? Like, knock yourselves out. So we teamed up with PF, sprayed it, plowed it up, planted it down with, flowers and different types of pollinator habitats. Boom, we got these great new food plots growing right adjacent to the highway. It's land that was excess. They didn't want to use it, and we're making use of it now for habitat, and it's going to work for both pollinators and, in the right places where if you've got, small game or pheasants and things like that, that can be used. So one of the things we're trying to do is expand that, looking at utility corridors, looking at riparian corridors, looking at other road, corridors where, especially things that go north and south, because we want to try to mimic and provide that type of habitat and milkweed for the monarchs as they migrate back and forth. So a lot of good potential there, and who's against butterflies? So to us, it's a really neat opportunity to reach out to some new and exciting partnerships, and I think demonstrate relevance from a wildlife management standpoint to a lot of folks that would never think of us otherwise. So, and then obviously recruitment, retention, reactivation is just a constant battle mantra that we're trying to do. We are doing some neat things that we're trying to think about. How do we do things, change the paradigm from some of the old, big one-day events, rather than doing that, trying to get in some mentoring type of programs and look at things long-term. Here's a really important thing for you college students that are thinking about, what am I going to do for the rest of my life? Career offerings, some of the things that we have on our staff. From a biologist standpoint, we have both fisheries and wildlife biologists. We also have fish and wildlife, what we call management technicians. These are the guys that you get out in the field and you get your fingers dirty, right? You're on the tractor, you're out doing habitat improvement and things like that. If you're more of a hands-on type rather than the scientific type, that's where those positions come in. Hey, hatchery production personnel, again, or as we call them, the fish squeezers. But you know what? That's important work. And quite frankly, some of our hatcheries are probably some of the most efficient in the nation. we actually had a performance audit done by the Auditor of State last year, and in all but one of the categories they looked at, in all but one of the species that we work with, we were more efficient and effective than private sector hatcheries in raising fish, especially when it comes to frying and fingerling. So that was pretty cool. Law enforcement, obviously, from a wildlife officer's job, we have an officer in all 88 counties. We also have investigators and some supervisory positions as well, and that's usually about every other or every 2 years. We'll do a cadet academy, looking for folks that are interested in helping out with law enforcement and information and education staff, which helps push the message out and provide the education, whether it's hunter ed, whether it's trapper ed, those types of things, step outside type of programs. Again, the cadets, we just went through a cadet class last year, started 16 new officers out of 800 applications. So the competition's pretty keen, but there's always opportunities that come. So that's kind of where I wanted to get and summarize about what we are, who we are, what we do, and I'll try to answer any questions you might have if I hopefully didn't put you all to sleep.
How are you, Zody? Thanks so much for coming today. What, if anything, can we do as hunters, anglers, and trappers? Any volunteer opportunities with the division here in Ohio?
Sure. Well, we have different types of opportunities for folks to volunteer. And it could be something as simple as going and helping out at a sports show or helping out at an education class, or going out to one of the wildlife areas if they're going to do a cleanup day. If you're looking for simple things like that, I think some of the bigger things we really need to focus on is having our current hunters and anglers help us create new hunters and anglers. So I think that the best thing they could do is be that big brother or big sister, quite frankly, type of person and mentor someone into one of those sports and help us be, a strong and viable organization. And that's one of the things we're trying to do is shift that paradigm. So instead of we're trying to get more of those one-on-one or small group type of activities going. Yes, sir.
Again, thank you for coming. I work at a little bait shop in Waldo, Ohio, and I've been hearing some stories. An ODNR officer actually came in and talked to us, and he said that we're starting to find some little baby long-nosed gar in some of the rivers in Ohio. I didn't know if that was true. I know they're pretty prominent in the South, but I never really knew they came up this far.
Yeah, actually, it's one of those quiet success stories that we are seeing some greater numbers of gar. Paddlefish down in the Ohio River are doing well and doing better. We've just delisted bobcats in Ohio because they are growing and thriving very well. And then this year, it was the last year we're actually going to be banding peregrine falcons because, again, they've done a really neat job to spread out and proliferated. And so we've got some neat success stories out there that we need to keep our horns about a little bit. So, here.
What was your, I'm sure you studied this, reiterating, what's the biggest issue with recruitment of new hunters? What's the underlying issue that you see not being in there?
Right there. We got to get people to step outside. That's the bottom line. Yes, sir.
Just a quick question about invasive species, like with Lake Erie. What strategy does the organization implement to combat zebra mussels and gobies? And has it been effective in getting rid of those?
Zebra mussels and gobies are pretty much here and here to stay. And you try to work on, quite frankly, managing and tolerating for those species. Now, I know that there's some experimental things going on with respect to zebra and quagga mussels that I think Michigan has tested on some inland waters that have showed some promise, but we are not we're not ready to put put that to work in Lake Erie without some more testing and making sure that there aren't other detrimental impacts. And it's, well, I won't go down the weeds on that, but suffice it to say, there are some things that are being worked on with respect to that. The main thing we're trying to do, like with Asian carp, is, try to close the door before they get here. The same thing with, like, snakeheads. People have heard of snakeheads. They're more in the eastern states, but that's something that we are try to keep an eye out for and prevent from coming in rather than worrying about them after they get here. So that'd be, a preventive that we're trying to work towards. And we work with our aquaculture industry, folks that raise fish for food and things like that, on trying to make sure that we don't have them inadvertently bring something in that could be detrimental either to native stocks or, like Asian carp, if it gets out and proliferates, you've got all sorts of different problems there. So I don't know if that answered your question or not. Okay. Yes, sir.
Thank you for coming. So you're talking about the North American model for wildlife conservation, and the second thing you mentioned was limited commercialization. I was just wondering if you had any comment, like, on the state of that goal in America, because I know you said the state isn't leasing any lands for shale gas drilling, but I know in other states, in the federal land, that's like not the case.
Right. Well, let me see, because you're kind of taking me down two different roads with one question. When you're talking about, and I guess with commercialization, when you're talking about the North American model, that's more in terms of commercializing the wildlife, i.e. commercial fishing or, taking a wildlife species and breeding it and using it for personal gain or private type of industry. But when we talk about leasing of minerals, at this point in time, the policy of the state of Ohio and this governor is, we're not ready to do that yet. That may change. I mean, I know that the mechanism is there in the law that allows it to happen. It's more of a policy matter that's decided above my pay grade. That's okay. But is it something that could be beneficial from a resource standpoint, from a funding standpoint, if it's done properly and done right? Yeah, absolutely. Pennsylvania is probably a good example of where that's been successful. Yes, sir.
I was wondering if you could touch a little bit on the white-tailed deer management strategy and division.
Okay.
I guess that's a really broad question, but
yes.
Is there any? Do you utilize any culling strategies or birth control strategies, or you mainly just rely on public hunting?
Hunting is the single most effective tool when it comes to managing white-tailed deer, because beyond human beings, the only predator they have is either a Ford, Chevy, or Dodge. So, sorry. I mean, but I'm just trying to be realistic here. But to your point, we recognize that, especially in some urban and suburban areas, where you can't get access or opportunity for hunting, then you have to resort to more creative measures. And so our biologists have worked with a lot of your cities and suburbs on whether it's culling through sharpshooting, whether it's controlled type of hunting programs, especially focused on archery. Archery hunting can be extremely effective in an urban environment. We've proven that in several locations throughout the state. And then, you're dealing with, the public, and half the people support it, half the people don't, and, that your leaders at the local level sometimes get caught in between in that tug of war. We do not recognize any birth control as an effective or viable means. Doesn't mean that maybe someday you might get there and you might find a proper environment to test it or allow it in, but there's still too many questions rather than knowns about things like that. We do have a pilot experiment going on right now, project down in a small neighborhood in Cincinnati that, due to its geographic location and I-75 and a few other physical features, is pretty much an isolated community, and we are allowing them to try a trap, sterilize, and release program and see how that goes. We've basically given them a permit to do that for 3 years, keep an eye on things, see what happens. If they still continue to grow their population down there, obviously then they're still getting migration in and out, and we'll probably say, okay, you need to employ some lethal methods in addition to what you're doing here, because this is not working because you're not isolated. So, Brett.
Chief Zody, could you maybe give our students some pointers or guidelines on how they might guide their educational plan, their pathway toward a career in natural resources?
Well, I know, and I say that, Fred, because there's so many pathways that are out there when you're talking about natural resources. Because there's, I think, a certain pathway for fish and wildlife management, which obviously is going to be that biological, zoological, and fish and wildlife management type of thing. But if you're, interested in forestry, there's courses in a pathway for that. Mining and mineral resources, oil and gas exploration. You've got petroleum engineering types of programs or geology. So honestly, I think a lot of it depends on where you think your passion is and what you think you want to do or get involved with. Or, if you're really not sure at this point, it's okay to be a generalist and zero in on things as you go. I guess that would be the best thing I would say if you're interested in a natural resources career. The one thing I will tell anybody, I don't care whether you're wanting to get into natural resources as a career or what your ambitions in life are, if you want to be successful and be satisfied with your career, figure out the things you love to do, and then find out a way to get somebody to pay you to do it. And I'm dead serious. That's the secret to success.
So given the audience that we have here, there's going to be a lot of biologists in this field. Is a terminal degree at this stage to get into a position like the DNR? Is it more the undergrad degree, or are you hiring a lot of master's or PhD students?
Gotcha. I say there's a mix. I think more today than, say, 10, 20, 30 years ago. I think that undergrad, that 4-year degree, is pretty important. We do still hire folks with just an associate's. It tends to be more along in the law enforcement or in some of the tech and more the hands-on type of positions. But I think especially if you're really interested in doing the research and the higher level things, that absolutely, I would encourage a minimum of the 4-year undergrad and even a master's, or start with us and work towards that master's as you go. But we've got our share of PhDs. I mean, both my dear biologists are PhDs. I'm trying to think if any of the fisheries guys are.
Well, you got Chris Vandergood.
Yeah, that's right, he is. So.
And then you're seeing a lot more of that. It's not just in the DNR, but EPA. A lot of the state agencies, if you want that position, that's likely a master's. Most of the positions, I think, is master's, but master's preferred, I think, is kind of some of the language that's in there. So just that's something you should know when you're going down that road that you may not be hearing from your counselor your freshman year, that you might need a master's degree. So just put it on your radar.
Yes.
Any other questions? So I actually wanted, go, please.
I was just, what's cool stuff do you see coming down, or legislation in the past, essentially something like, a couple years ago, Greenwald rifle cartridges are going to be, now he was talking deer management from that perspective, but are potentially like trophy deer management perspective.
Actually, one of the things we're looking towards implementing, I don't know if we'll get to it next year, but maybe the following year, is going, instead of doing county by county deer management bag limits, we're going to what we call deer management units, where we're trying to lump together similar habitat, similar geography, and similar deer mechanics, if you will, and manage on those levels. So it's going to, we're working on the draft zones right now, and it's going to be a different concept for our hunters to swallow, but I think once they get used to it, it's going to make our management decisions a little better and a little more informed. We actually have a study being done right now by Southwick and Associates, which is they're a private company, but their basically core business is working with state fish and wildlife and natural resource agencies on financial and license and permit types of questions. And they're looking at our entire license and permit structure from a cost standpoint, from an availability standpoint, and from a business model standpoint. Does it make sense? Should we be looking at some type of a hunting fishing combo, or a deer hunter package, or a sportsman's package, things like that. So we should be getting some recommendations from them later this year, and that would be something that we definitely need to take down to the legislatures and state house, because we'd have to, if they're recommending things to make changes, that have to be done in the code rather than by rule. So.
And one of the things that I want to mention before we let Chief Zody go is that many people, I think, wrongly see the DNR as an agency that's kind of isolated from other things that are going on. And you need to know that they're fantastic at partnering and bringing other programs into the fold. A lot of the research that's going on is reaching out with things like Pheasants Forever and Ducks Unlimited and the Nature Conservancy and research entities. And so just for, in my relationship in the time that I've been here as the interim director, I know that we've got funding that's going to you to look at the Ballville Dam coming down and tracking walleye movements. I know that Tori is teaching the fisheries class that's going on right now, or the fishing course that's going on right now, has been out on the vessel and has put underwater cameras down with DNR staff members to see how the nets are deployed and how those instruments are catching fish. The Aquatic Visitor Center is a great resource for Ohio Sea Grant Stone Lab to educate the general public, and that wouldn't happen without the generous support of the DNR. And so you really need to know that the DNR and the Division of Wildlife is there to work with stakeholders and to work with other programs and agencies. One of the greatest examples I can think of is the LEPMEG project. That's LEPMEG, and basically what this is, LEPMEG, it's the Lake Erie Perkit, or the perch management, and basically helping to set what the quotas are for harvesting fish. And what's been happening, and I would say in the last 7 to 10 years, is you brought in the stakeholders, you brought in the charter captains, and actually opened up the door and say, how do we determine how many walleye you can take? And so they're really trying to show that this is a complex formula that goes into determining that. And we're not just grabbing numbers from there. And they're showing the math and the statistics that go into that. And you're really seeing the charter captains realize that they're not trying to limit our success as a private business owner. They're trying to sustain our program. All right, if you randomly grab numbers, well, then one year the harvest might be high and there's lots of fish out there, but then it could crash the next year and your business crashes with it. And so they're realizing that the regulations that are put in place are really to protect the resource, but also to protect the stakeholder in the industry. And so I'm really excited to work with the DNR as we go forward, because the partnerships and the projects that are coming out of that agency are fantastic.
I want to piggyback on what Chris is saying. I'm Lindsay Manza, one of the instructors for the educators this week, and 3 of the 5 curricula that the teachers that are up here are learning and using are all provided free of charge by Division of Wildlife through Jen Dennison's office. So Project Wild, Wild Aquatic, and Science Civics is all of the curricula that they are learning to take back, right back into a classroom or back into the informal setting. So it's another example. It's not so much a partnership, but we work very closely with Jen to do the training to make sure that we're implementing the curricula that she has, and then we're doing it here at the grant.
Thank you.
Great. Chief Zody, thank you for the visit, and if we can give a round of applause. So, wait for Kevin here. So, of course, this happens every Thursday. Again, you guys will be departing from our island, sadly, on Saturday. Good luck on those exams coming up. Go quickly. Actually, I didn't ask at the beginning, any of the professors, do you have any announcements that you want to make, Lindsay or Angie? Tori or Fred, any announcements for your students? You good? Justin Chaplin or Jeremy Pinkerton, any announcements for your students?
It's a big day tomorrow.
Good. Test. Good. So when you do depart from the island, hopefully we'll see you again later this summer, if not next summer. But next Thursday we'll have another set of lectures. So our guest lecturer will be Jim McCormick. So he's going to speak about some of the bird populations in Ohio. And then our research brief is James Marshall. He actually teaches our our evolution class here on the island, but he'll be talking about bird research too. So please go to our website, get on the webinar, and listen to these virtually. And thanks for your attendance tonight.